Welcome, and thank you so much for joining today's session, Frequently Asked Questions from the International Myeloma Society annual meeting. I'm Quinn Young, Chief Mission Officer at the Multiple Myeloma Research Foundation, and I am so privileged to be joined by Dr. Tom Martins from UCSF in San Francisco, California, and Dr. Krina Patel from MD Anderson Cancer Center in Houston, Texas. We've invited them here today because we had so many questions coming out of our highlights from the International Myeloma Society webinar last month, so that they could answer some of those questions that we weren't able to get to during the session. So without further delay, let's get started. So starting with newly diagnosed multiple myeloma, we've seen during the meeting, we saw updated results from a number of clinical trials, in my opinion, basically reinforcing the effectiveness of quad therapies. So, overall, starting with you, Dr. Martin, what would you say the biggest takeaway was from that meeting for patients?
So in the last 2 years, we've had a number of presentations on quad-based therapy, and almost all randomized trials that we've done in myeloma, more is always better. For whatever reason, more seems better. And for this one, actually, when we add quad therapy, which typically is a CD38 antibody to our backbone of VRD, bortezomib, lenalidomide, dexamethasone, you see an improvement in overall response rate. You see an improvement in progression-free survival, and also in MRD negative levels. And we think in time we might actually see an overall survival advantage. We'll see. It's just too early for follow-up. But so each meeting, we do get some follow-ups from some of the studies. One of the big ones that we have had results from is called the Perseus study. And that was DARA with VRD versus VRD. And they did a subset analysis of patients who had high risk and showed that, in fact, the quadruplet-based therapy is a beneficial therapy for patients who have high-risk disease. And there's always been a question of, does the CD38 in newly diagnosed, does it really help with, the high-risk group? And I think we all are convinced now that it is a component that helps. I think one of the big questions still is, for everybody, is quad-based therapy. The way they're done for transplant eligible is quad therapy, transplant, a couple cycles of quad therapy, and then maintenance. And that maintenance can sometimes be a singlet, just Revlimid, or it can be a doublet, Revlimid plus daratumumab. Well, with high risk, and I'll talk about that in a second, we always go with more for maintenance. And so in high risk, I think the daratumumab plus lenalidomide is probably the best thing to do, more than just one drug for maintenance. And in the high risk group of patients, we saw an update also from what was called the GMMG concept study, and that's from Germany. And that was actually using a different triplet, the triplet of KRD, so carfilzomib, lenalidomide, dexamethasone, in patients who had high-risk disease. And it actually showed one of the best progression-free survivals in the truly high-risk people. And I'll point out the differences in that study. And that is, in that study, the patients got quadruplet induction, they got transplant, they got quadruplet consolidation, 4 cycles of quadruplet, which was, this one was basically isatuximab, carfilzomib, lenalidomide, dexamethasone. And then they got continuous therapy for 26 cycles. So for 2 years, they got triplet-based maintenance. There was ISA, carfilzomib, and lenalidomide. So ISA KR. And I do think with high risk, and we'll see what Dr. Patel thinks also, that with high risk, my practice is to use more rather than less in high risk. And I usually do use 3 drugs, not 2 drugs for maintenance for a long period of time. That said, I'll just say just a small thing about the transplant ineligible population. We had 2, nice publications that were presented at ASCO, EHA, and then some updates in this meeting. We had a nice session on induction therapy. And these were for transplant ineligible, also looking at quad versus triplet. In one study, the quad was basically isatuximab VRD, and the triplet was VRD. The other study, it was ISA-RD versus ISA-VRD. So it was a change that, was the bortezomib needed? Typically, we say is the CD38 needed. This one was the bortezomib. It's called the benefit study. And just like every other study, the quad always wins out. So the quad won. And so for patients who are not intending to go to transplant, these included patients up to age 80, as long as they're in good shape and they're fit, they can get a quadruplet-based therapy, in my mind, with no added toxicity. And I think that's really should be standard of care. The big thing in the benefit study, which I really liked, was it was bortezomib or no bortezomib. But in the arm, the quad that had bortezomib in it, it was weekly bortezomib, not twice weekly. So weekly, a big deal, big difference. And it was 12 months of weekly. And then they went an extra 6 months of every other week dosing of the bortezomib. So it's a longer bortezomib overall, but less because it was just once a week rather than twice a week. And there was less neuropathy. That's, I think, what most of us have adopted as our standard of care for that population. So in general, really great data. In the big picture of things, if you take transplant ineligible and you give them one of these quads, we think the remission duration is going to be 7 to 8 years. That means one line of therapy, nothing else. 7 to 8 years they're going to be in remission. That's better than sometimes, 5 years ago, the overall survival was for people. This is just first line of therapy, PFS. And in the transplant eligible, where you get a transplant and then get these drugs, I heard some crazy numbers pushed out, 12, 10, 12 or years or longer of your remission duration. So just fantastic results. So that's really the take home message.
So a couple of things, even before Dr. Patel, we get your take on the question, a couple of definitions to level set for the audience. One is definition of high risk, and then two, transplant ineligible, because there's the transplant ineligible definition in a lot of these studies, particularly ones say, that are global, versus what does it really mean to be transplant ineligible? So if we start with high risk, what's the definition of high risk? And knowing that for our audience, this is also evolving and rapidly so.
Yeah, so I think, as Dr. Martin said, we are doing so much better with quadruplets, but really there's still a group of patients, about 20% of our patients, who unfortunately don't get that long-term PFS, that 8, 9 years if they're not transplant eligible, or the 12 plus years if they are transplant eligible. So really it's who are the patients, what are the biomarkers we can look at that tell us ahead of time who these patients are, so we can decide they should get more maintenance therapy, again, as Dr. Martin said. So right now we have cytogenetic risks as one of the definitions. And so classically, we've always said someone who has 17p deletion, right? So some of these chromosomal abnormalities, something called, translocation 414 or 1416. And then in the last few years, maybe last decade, we added something called 1q amplification. We added something called 1p deletion, but none of us really knew, does that mean anybody with this, or does it mean you have to have a certain percent? And so really at IMS, at our big IMWG meeting in, I think, 2023, there was a consensus that was guidelines that were built to say, hey, maybe we should look at this data and say, who are those patients? Who are those 20% of patients that get less than 3 years, that really don't do as well because their biology is just different. And so it ended up being that we probably need some combinations. So maybe the 17p, you need at least 20% of your cells to have that, rather than even 1% counting. And maybe with 1q or the 1p deletion, you need 2 knockouts of that, or you need the 1p plus the 1q. So it's a little complicated, but it really is looking at those cytogenetics to say, okay, at diagnosis, who can I tell has high-risk disease, so that I need to do something different than what we do for everybody else. And I think the other, we talk about high risk in terms of disease biology without the cytogenetics. So people that have a high beta-2 microglobulin, which technically is just a non-specific, inflammatory marker, but if you have renal failure, it's going to be high automatically. So what they're changing is that if you have normal kidney function and then you have a high beta-2 microglobulin, well, that tells us more about the myeloma biology, and maybe those patients really are at a higher risk. So, there's different ways we're looking at it. There's also a bucket of patients that we think have normal risk and they're okay, but they unfortunately still relapse a lot earlier than what we expect them to. So we would still call that high risk, in that second line, and really, again, trying to get our novel therapies to those patients so that we can catch up and get those, decades of response instead.
And then tackling transplant ineligible. What is, how it's defined in studies versus, I would say, in reality in the U.S.
Yeah, so in the US is where we make it harder. I think Europe and everywhere else, they really have age cutoff and they have, certain organ cutoff. But for us, really, there's a lot of patients who are transplant deferred because they don't want to go to transplant. And we don't have an age because age really is just a number for us. It goes down to performance status. I will say clinically, there's two main things. If someone has significant heart failure or if someone has significant pulmonary COPD type things, those are the two organs that make them a little bit higher risk of infections and sort of worse outcomes for the transplant. But even patients on dialysis, et cetera, we can get through transplant. I think something that's become a little bit more controversial in the last few years is age. So even though I just said age is just a number, sometimes, patients who are above 70 or definitely above 75 will get a lower dose of chemo, that melphalan, which again, there's some studies say that's okay and others that say maybe you're not going to do as well with that lower dose. It's not going to help your myeloma as well. So again, there's a little bit of controversy still, but I think for the most part, you're truly eligible if as long as your heart and lungs are really working well.
Our friend and colleague Nina Shah used to give it the Target test, meaning if you can go to Target and you can take the shopping cart around and put stuff in and check out and get back to your car, you're transplant eligible.
Oh, there you go.
Yeah. No, we try in communicating to patients to incorporate that element of choice as well, because there aren't those same cutoffs as you said as there are in Europe. So the concept of transplant eligible ineligible doesn't seem to be as applicable here. So I think sometimes the reporting of results from studies like that are a little bit confusing for patients in the U.S. So before we move on to building on some of the discussions around minimal residual disease, which came up a little bit in the results from some of those frontline trials, let's discuss a little bit about what's coming, the CELMoDs, BlenRep. There was some interesting frontline and maintenance data presented with those agents. Maybe each of you could take one of those and highlight again what could be coming down the road for patients.
All right. So I'll take the easy one. I'll take iberdomide. Krina, you'd have a hard one. So iberdomide is a CELMoD, and basically it's a next generation lenalidomide or pomalidomide. It has the same target. The target is basically to degrade a couple of the proteins that exist in the cell that makes the cell grow and basically stay alive. And so they're called Ikaros and Aielos. And by degrading those, basically the cell then undergoes apoptosis and dies. But they have the CELMoDs and also lenalidomide, pomalidomide have another effect, and that is their pro-immune system. They actually stimulate the immune system. And iberdomide does it with much more potency than either lenalidomide or pomalidomide. So it stimulates the immune system more, and it also basically can kill off more myeloma cells, even myeloma cells that are resistant to lenalidomide or Revlimid or pomalidomide, Pomalyst. If they're resistant to those drugs, you still can get a response. 25% will still respond with iberdomide. So all the drugs that we test in the late line and we think they're working really well, better than what we had expected, we try to bring them in earlier lines of therapy. And so one of the possibilities for iberdomide is to use it as maintenance post-transplant. And so Tanya Wiles actually presented some maintenance data that, in fact, iberdomide is probably better tolerated, also less side effects than lenalidomide as maintenance-based therapy. I think everybody knows that lenalidomide has associated fatigue, diarrhea, and lowering of the blood counts, which can be problematic over time. And in fact, after a year, at least a third of the patients come off of maintenance. After 2 years, maybe 2/3 of the patients come off of maintenance-based therapy. And so iberdomide, actually, you have less fatigue. I think you have way less GI side effects. And maybe the only thing is count suppression, and that can be mitigated with dose reductions and dose alterations. So this is in a randomized trial right now, and we're looking forward to see the results of that randomized trial. The hard part is half of us might be retired by the time those randomized trials are ready to go. But I do think it's going to be one that we could use. It's also being tested in transplant ineligible. We've seen data previously with iberdomide plus bortezomib in dexamethasone in frontline, but we are now seeing data with iberdomide plus daratumumab in dexamethasone. So the standard, one of the standard triplets for transplant eligible is daratumumab, lenalidomide and dex. So this is daratumumab, iberdomide and dex, probably maybe a better lenalidomide, right? And so the initial data is suggesting very high response rates. We do have to, follow these patients up over a longer period of time. But if I had my crystal ball in front of me, I do think that this combination is going to be more potent than DRD. I think DID, Iber, Dex is going to be better over time. I think we're going to come to that conclusion. And it might be a really well-tolerated triplet-based therapy, especially for older patients, maybe patients who cannot tolerate a quadruplet. So very exciting data, actually. Your turn, Krina.
Yeah, I can take over. Again, BCMA, which is a great target for myeloma. We have lots of CAR Ts and we talk about bispecifics all the time, but we have our ADC too, our antibody drug conjugate that's stuck to that BCMA. So it can take chemo basically to the myeloma cells. And then we talked about DREAM 7 and 8 the last couple of years, last few conferences we've had where it was an early relapse. But now they actually just recently presented a frontline trial, which is kind of exciting, where it's VRD plus Bela. So, again, not using a CD38, but a BCMA specific ADC. And again, not a monoclonal antibody necessarily. So these were patients who were transplant eligible. They ended up getting Bela VRD for 4 cycles as induction, would go to transplant and then go to maintenance. And what was interesting, what they've learned from the previous study is that when you give Bela less often, you can still get a great response, but you decrease the eye toxicities. And I think that's what they're able to show, that, in the induction, still 40% of patients or so had decrease in ocular AEs or had a decrease in vision, but that improved post-transplant. And then during maintenance, I think only 1 patient at the end of the 1 year that they're looking at for the study actually had any kind of vision deficit still. So over time, it does improve, and giving it, every 8 weeks is better than giving it every 4 weeks. So I think, again, toxicity-wise, everything else, in terms of hematological, your counts, et cetera, they were actually pretty well tolerated and no major issues. And I think where this comes in as an important topic is, with our bispecifics and CAR Ts, which we all love, but there's a lot of risk of infections and high-grade infections. And here, we're not really seeing it to that level. But again, the compromise is your eyes. So I think, we're learning how to use these in different patient populations as well.
And another potential advantage of both the Belmaf and the cell mods like iberdomide is they're off the shelf. So for patients who can't get to an academic center and be hospitalized either for the CAR T or even for the initial doses of the bispecifics, those could present good options, for certain patient populations, given for their convenience. So I do want to circle back to the concept of MRD. And on the webinar, there were tons of questions because we were talking about MRD, like everyone understood what MRD is and how you measure it and what the thresholds are. So before we talk about dive a little bit deeper into some of the clinical results and where we saw these incredible rates of MRD negativity and what that could mean for prognosis, Dr. Patel, let's just start with what does MRD mean? And how is that testing done? And what are the different thresholds to measure it?
Yeah, so I think it's really important. We talk about response with overall response, partial response, all that kind of stuff, which I always tell my patients that a stringent, complete response is the best response you can get based on your blood work and the proteins in your blood and your urine that we always have you come send to us, as well as your bone marrow based on just the stains that the pathologist does. But if you really want to look, are there any other myeloma cells below that superficial level of the bone marrow? That's where MRD comes in. So that minimal residual disease is what it stands for. And I call it undetectable rather than negative, because again, The deeper, the lower, like we talk about 10 to the minus 5th level versus 10 to the minus 6th level. We're trying to get to 10 to the minus 7 eventually. The deeper that is, the more likely there's less myeloma cells in that bone marrow, right? And it's just depending on our technology of how deep we can get to tell you how reliable it is. So again, the deeper, the better. But we know that our patients who can get to 10 to the minus 5th, and this is from bone marrow right now. So we are working on blood tests for the future, and these are in research, but hopefully will become a standard of care in the future. But right now, when we do your bone marrows, we can actually take some of it and send it to either a company called Adaptive that they do next generation sequencing, and they actually look at your original bone marrow and look at the clones that you had, and they're able to track to see if they find any other ones in the, once you have a response, and they can tell us, to the level of the cells, like what number that they still see that have some of your myeloma clones in there. And those are myeloma cells. So again, if we can get to 10 to the minus 5th or better minus 6, we know prognostically that patients are going to do much better for a lot longer. They're the ones who are going to get that progression-free survival of, years or even decades. And then we know, especially for, you have to put it in context of high risk versus standard risk disease though. So everything I'm saying right now really makes sense for standard risk. For high risk, we still have to make sure that long-term they stay in that same MRD undetectable. So that sustained MRD is really important for all our patients, but especially our high risk, that not just a one-time point, we got to see it over time. And I think what it's helped us with is the FDA, number one, we can actually now use this in our clinical trials to try to get new therapies approved earlier rather than waiting years to see which one works better, which is huge. But the second part is, in clinical trials too, can we potentially stop therapy for patients who are in MRD undetectable state for a while? Can we stop over-treating patients? Make sure that high-risk patients still get treated, but how about we stop it for patients who might not need it anymore? So I think, again, there's a lot of functional ways we're going to be able to use MRD going forward.
And one question came in, you mentioned we need to use bone marrow. One question came in from a particularly astute listener in terms of using mass spec with blood and where we might be with that.
Yeah, so I haven't been using it. We don't have it, and we tried to get this at our institution, but we're still with the light chains and all those things, and then just using imaging and PET scan. So I think mass spec is great, and I do think that Mayo uses it a lot, and we'll see if Dr. Martin uses it at all. But it's just a different, deeper way to look at proteins and look for myeloma in the blood. I do think that all these different tests that we have, so there's something called CTC, circulating tumor cells, or circulating DNA. There's so many different ways we can look at it, and really, I think in the end, it's how can I tell my patient who might have bone marrow disease, who might have extramedullary disease or disease in their, either skeletal or outside the skeleton, plus, anything else that we might find. How can I use one test to find it all? And I think we're still a little bit far from there, but it's really a combination of these that can get us that deeper level, compared to the studies we've had classically, that can really tell us if our patients are under, their disease is under control or not.
So I'm going to add to that also. I completely agree, and that is, the mass spec machines are needed for this. We're hoping that they're going to be put in place around the country. It'll be a send out lab for all of us, and hopefully by the end of next year we'll be able to do it for majority of people. And if you just think of it, when people are first diagnosed, they have a fair amount of protein, and we're just watching the protein go down in their blood, go down and down until it's not detectable anymore. And when that protein in the blood is not detectable anymore, then we do the bone marrow to look for MRD, because there's still likely a small number of myeloma cells in the bone marrow. But with mass spec, it's going to drop that level of detection down to even a lower level, so that we won't have to do a bone marrow biopsy as early to look for MRD in the bone marrow, because we still be able to detect that protein in the blood. So it'll save on the number of bone marrows that we have to do for sure. But I do think they're going to work in concert, that once we get that mass spec undetectable, then we'll do the bone marrow to look for bone marrow MRD. Maybe at that point in time, we're able to do peripheral blood looking at circulating DNA, like Krina just said, to do that. When people get their MRD results, because it's quite complicated to read them. In fact, it's quite complicated for the doctors and a lot of the referral docs to read them also. It's kind of a little crazy, but just in general, one cell in 100,000 is 10 to the minus fifth. One cell in a million is 10 to the minus six. You want less than that, right? So when you get that thing from Sequenta and it says 9, we've seen 9 cells per million, that's 9 per million or less than 1 per 100,000. That's MRD negative. So up to 10 is MRD negative for the Sequenta. The one, if you do flow cytometry, it usually gives you a percentage, and you can just convert that into, is it more than 1 in 100,000 or less? That's your MRD level at the current time. It is complicated, but hopefully you'll as you look at them, they'll become less complicated.
So, and Dr. Patel, you mentioned incorporating MRD negativity as a measurement into clinical trials, and FDA did recognize that earlier this year. We were all very happy about that as an intermediate endpoint. What about MRD negativity in outside of a clinical trial, in clinical practice? And at what point do we think we may be able to say if a patient's MRD negative for a certain amount of time, they could be cured?
Yeah, that's a great question. I want it to be now, but I can't say that yet, right? I will say the closest we've gotten to a functional cure has been in the CAR T one and done, where people are not on treatment, and those patients who are 5 years out and have done really well. We just haven't had enough patients in that category yet to say, okay, how can we use that MRD testing? I do think there are a couple of clinical trials that were presented at IMS that looked at, can we stop therapy, right? Which is, I think, a bigger question for a lot of my patients that are on maintenance therapy, either after transplant or induction. And so there are a couple of trials that looked at that. One looked at patients who are on LUN maintenance for at least 3 years in a CR. And if you're MRD undetectable, let's stop maintenance and see what happens versus keep going. And I think it was a small number of patients, but did show that maybe those patients are going to do just as well. Again, we need bigger studies to validate that. But that's what I do in my clinic. If I have a standard risk patient who's gotten through at least for 3 or 4 years of LUN maintenance and are having any major side effects, or even side effects that are just slowly getting worse, right? Their counts or things they're not going as well. And they ask me, can I stop? We do try to use MRD to help us. Now, if someone has really bad side effects, we stop anyway, right? Quality of life matters as well. But it's really those patients who are doing okay, and they're just wondering, should I keep going or not? We will use MRD to help us with that. And I think, there's also been studies saying, do we need to do transplant or not? Again, I'm not there saying that we don't need transplant yet, but there are studies that are looking at that. And one of them, there was a readout at IMS where maybe if you get, MRD undetectable, you don't need to go to transplant. We can defer it. And so again, we need longer term data to say that that is the right choice or not for our standard risk patients. But again, really interesting to hopefully help us say that, yeah, that's the right thing to do. And right now in clinic, if I have a patient who really doesn't want to go to transplant, we talk about it and we still will use that MRD as part of that decision just to see. But I don't just automatically say you don't go to transplant or you do based on that MRD.
Yeah, it is part of the decision tree. It's not the whole decision tree. It's just part of it. Joaquin Martinez Lopez from Spain, who's done a lot of publications on MRD, likes to come for San Francisco. He usually comes every summer, and he goes through all our transplant data and our MRD data because we just do it routinely on everybody. And he published a study a few years ago, over 400 patients looking at, do we actually change therapy based on MRD? Because all of us said, of course not. It's not ready for prime time. But in fact, he said about a third of the time, you guys make a change based on the MRD. Mm-hmm. And most of the time, the MRD change or the MRD driven change was to take medicines away because they're MRD negative and they're having side effects. So we took their medicine away. And then he looked at the progression-free survival curves for the people who had MRD changed versus those that didn't have MRD changed for whatever reason. And honestly, the ones that had it, even though we took the medicine away, the people that we made a change did better in general than the people we made no change. And it's just data to suggest that we probably can use this in the future as a way to modify treatment based on results.
So one final question in the upfront setting before we move to relapsed refractory. Someone wrote in, what about a 5-drug upfront combination?
Well, there is a 5-drug combination actually from the UK, and that is specifically in the high-risk patients, and they added cyclophosphamide into the DARA VRD. And honestly, the data looks really quite good, but they also not only did 5 drugs, a transplant, and then 4 drugs, prolonged 4 drugs, and then 3 drugs forever. It's hard to know what component of it really helps. I will tell you that the 5 drug with cyclophosphamide is not the 5th drug that I want, right? The 5th drug, if I'm going to add one in, is one of these newer ones, a BCMA targeted drug, right? And so I would say hold the phone on that question, because we're going to have trials like that. But the truth is the BCMA targeted therapies, when we bring them up front, they may be potent enough that we don't need 5 drugs. We only may need 2 drugs or 3 drugs. We might not even need a quad. We just need a doublet or a triplet, and it'd be as good or better than the quad because they're so powerful. That's what I'm hoping for.
Yeah. We have a trial using a BCMA bispecific as a single agent. And so hopefully we'll have data on that in the future. But I agree, you might have better potent drugs that you actually don't need multiple anymore.
So shifting gears a bit to relapse refractory and starting with CAR T, because that now in the US at least can be used in earlier relapse versus now the currently outside of clinical trials, the bispecifics are more for later relapse. Let's start. Dr. Martin, what would you say some of the high-level take-home messages around at least the approved CAR Ts in the US, Abecma and Carvykti, were coming out of IMS?
Okay, so with the newest studies from the basically both ide-cel and cilta-cel, the randomized phase 3 studies, the ones that led to full approval of these drugs, were both done in earlier lines of therapy. For cilta-cel, it was done in patients that had, progressed on lenalidomide. So these patients are lenalidomide refractory. So the approval is for patients that have lenalidomide refractory disease. That's still a tough patient population in general. But if you look at that's CARTITUDE-4, if you look at CARTITUDE-4 and you compare the CAR T as the therapy versus a standard triplet that we would have used in that line of therapy, hands down, the CAR T cell therapy works and is better. Now, we are finding that there's side effects from, the CAR T cell therapy, 2 important ones. One is the late neurologic toxicity, which was about 2% of Parkinson's-like syndrome and 10% cranial neuropathies in the CARTITUDE study. And there's also secondary cancers. So other cancers, myeloid malignancies that, again, around 1 to 2% in the CAR T cell, and other mostly skin cancers, and then some other solid tumor cancers. So these are big signals that we have to follow up and look at. And so, there is some debate in the myeloma community. Are these side effects, toxicities, too severe to bring these therapies in at least the first relapse and in the earlier lines of therapy? Well, I'm going to go on record in this conference right now and saying, I think that's all hogwash. All righty. Yes, of course we should use them as early as possible. It's the best therapy out there. And people try to compare the progression-free survival curve of CARTITUDE-4 with CARTITUDE-1, which it seems to just be a little bit above it. But that progression-free survival curve that we're all seeing is generally in the patients that have intent to treat outcome, meaning 15% of the people didn't get the CAR. If you compare those who got the CAR in CARTITUDE-4 versus those in CARTITUDE-1, there is a difference. But I think the biggest difference after CAR T is you're off therapy right now. It's the quality of life. I just saw a patient just before coming in the room. The patient I just saw is 2 years after his frontline CAR T cell therapy and saying, I feel fine. I feel normal. I feel like I did before I had multiple myeloma. And that's what we're hoping when we move these drugs in earlier lines of therapy. I'll also say for ide-cel, the data isn't as good in terms of progression-free survival, but it's because the patients in that study had triple class exposed multiple myeloma. They had a much tougher form of myeloma, and a lot of them were triple class refractory. We have a study through the BMT-CTN, and I'm curious to see what Krina thinks. For patients who got an autologous transplant but still had a measurable protein, and then we used ide-cel as the CAR T cell therapy. those patients are doing extremely well as a mop-up of minimal residual disease. I still think there's a role for all CARs in multiple myeloma. So I'm very encouraged about it. I think we still need to keep moving it as early as possible. Krina, what do you think?
Yeah, no, that's fantastic. I agree with you. I think you have two CAR T enthusiasts on right now, so I'm not going to debate you on that. We think the same. You know, I do think the side effects are something we talk about, we watch out for, and we make sure we can minimize, right? So we know that if patients go in with less myeloma, their bridging therapy is actually works, and they have less myeloma going in, their risk of a lot of these things is a lot lower too. Not just the CRS and regular neurotox, but the delayed Parkinsonianism especially. They've proven in multiple, different trials. And when people go early, I can actually give bridging that works. That's the other big thing. You know, I don't always talk about it as logistics, because I do think the quality of life piece, I think the progression-free survival, I think all of it is it's going to be much better in early line. But just the fact that I can give you bridging that works. And again, knowing that my patients are going in with the VGPR or even a CR sometimes in the real world, those are my patients that have done the best, the longest. So, I think we've already proven that yes, you need some myeloma, but probably not very much. It could be very minimal, and you're actually going to do really well. And kind of going in that, post even transplant cohort 2C of KarMMa 2, those are the patients that have done the best. They actually did really well when they went in with minimal disease. So I completely agree that going early is the best option to get patients to their cells, but also to have the best PFS and quality of life for the longest.
We might actually end up, the biggest risk to CAR T in my mind is maybe that the bispecifics, like Krina said, they have a frontline study. The bispecifics might be all you need, and you might not need a CAR T right away in the frontline. That would be awesome. But I think a lot of us are thinking at the cure of myeloma to be a combination of both, that it will be bispecifics plus CAR T cell therapy. And like Krina said, when people go into CAR with really minimal residual disease, maybe these side effects are much less. That maybe they won't occur at all. We'll see how that goes. And we have other new CARs coming down the pipe too.
Well, it is, both of you touched upon this a bit. There are very different results in terms of the progression-free survival until the disease comes back for patients, depending on a number of different factors when they get CAR T, because certainly the early trials where patients had seen 6, 7, 9 lines of therapy, the results were lower. So can you just explain a little bit more in terms of, the differences between the earlier trials of the patients who had CAR T much later in their disease versus what we're now seeing when it's used earlier, and what those key indicators for, okay, this patient is likely to get a really great long PFS coming out of CAR T?
So I think, some of the risks going into CAR T, no matter when, but especially later line, is that those people are more likely, if you wait till a long time, to have extramedullary disease, to have clonal escape, something that Krina brought up earlier, where that clone now has a 17p deletion, or has 2 or 3 fish or genetic abnormalities that make it really a tougher clone, and also that you can't bridge them accordingly, so they go in with bulky disease. Those are the things I think you take at risk if you wait till later on in the therapy, using that later on in therapy. So it's mostly lines of therapy. The median lines of therapy in that late CARTITUDE-1 was 6. In CARTITUDE-1, they basically had 1 to 2 lines of therapy. That's it. So that's great. Actually, it's a lot earlier.
Yeah, I think the patients that went on to the trials were different, right? So we know that not everybody can make it to 5th, 6th line. And we've seen that even in early line. So look at CARTITUDE-4, there was a big drop off at the beginning where patients never even made it to their cells because their disease was just so bad. We couldn't get it controlled to, get their cells, which again, the patients who made it to CARTITUDE-1, they probably had disease that was at least controllable. I'm not saying they didn't have high risk disease. Some of those patients did have EMD, but they were able to make it to their cells. So it's a different patient population when you're looking at that. And again, I think the way that they had to do the PFS, because intention to treat, meaning anyone that got on the trial, if they didn't get cells, is what that PFS, why it looks not as high as CARTITUDE-1 did for those really relapsed refractory patients. But I will say a lot of us that did these early line trials, we still put our high risk patients on these studies because we knew they weren't going to get to third, fourth, fifth line for CAR T. So when we have access, we give it to our patients that need it, right? So again, it is a completely different patient population, I think, in the trials too.
We touched a little bit more, Dr. Martin, earlier on about some of the side effects before you said it was all hogwash. To help patients understand, because there were a lot of questions when we did the webinar around, what exactly are those neurologic or Parkinsonian type side effects? And then I think what's really important too for patients is to understand when they may occur, just so that they go into CAR T, say, with more appropriate expectations.
Yeah, when you look at the results from CAR T cell therapies, they describe neurologic toxicities that are acute, that happen when the patients are in the hospital. And that's typically the most common one is headache. And this other thing called ICANS, where they can be a little bit confused. Those things are relatively easy to treat for us. Basically treating the fevers, treating CRS takes a lot of that away. And if they have ICANS, giving a low dose of steroids usually takes that away quite readily. And everybody basically gets better from those. But then about 3 weeks to 3 months afterwards, there are these delayed neurologic toxicities. The 3 delayed neurologic toxicities that we look out for, one is called the cranial neuropathies. It's the nerves that basically in the face and the upper above the neck, basically. And the typical ones are cranial nerve 7. So they give what's called a Bell's palsy, where the face can kind of swoop down on one side, and the eye, you have a hard time closing it, and people can have a little drooling from one side, or it can also be bilateral. Or the cranial nerve 3, where the patients can have double vision, where one eye doesn't go all the way to the side like it should. And so you end up getting double vision when you look to the sides. That happens about 10% of the time. The soonest we know about that, the easier it is to treat. We start steroids right away when we get those signs. And it's important for the caregiver to look for signs like that. If they see that there's it's unequal in their smile, it's unequal when they close their eyes, that's something we want to know about right away. And then there's peripheral neuropathy that sometimes is sensory, where you can get worsening sensory neuropathy, or worsening motor strength. And at worst, it can be Guillain-Barré, where it's actually fulminant, where people have a hard time really and are bedbound or basically in the hospital. That's less than 1% of the time. That doesn't happen all that often. And we've actually had a couple patients here, and we've turned them around with some steroids. And those things take a while. I say turn them around, but it takes a while and a lot of work on the patient's part to actually turn that around, a lot of physical therapy, et cetera, but it is reversible. The hardest one is the Parkinsonian features, where that people can have shuffling gait or really rigidity, or they have an attitude or an affect change. They're not participating in the communication with the family as they did before. Those are things the person just seems off. Those are things we want to know about too. And those are a little bit less, I would say, less responsive to some of our therapies, like steroids. Very, very often doesn't respond. And if it's true Parkinsonian with rigidity, et cetera, it doesn't respond all that well to Parkinsonian meds either. If their lymphocyte count's high in the blood, we try sometimes some therapies to try to we think that equate that with CARs. There's a lot of CARs around. We try to give them chemotherapy, try to get rid of the CARs, whether it's chemo through the veins or hydrosteroid through the veins or chemo into the spinal fluid to try to get rid of CARs that are around the spinal cord or the brain. We don't know what the best therapy is. We just are working on that out, but that also is low. It's 1 to 2% of the time at the current time.
So shifting to the bispecific, and we could spend a lot more than 15 minutes on the bispecifics, but there's the 2 groups, the BCMA targeting bispecifics, and then others like the GPRC5D, TALB. So Dr. Patel, can you briefly summarize, and they're typically used outside of clinical trials in later lines of therapy, but certainly a lot of clinical trials are, as we've discussed, looking at them in earlier lines. Basically give listeners a summary of what the bispecifics are, and particularly how they're different from CAR T, both in terms of administration, but also in terms of some of the side effects.
Yeah, no, I think the bispecifics are fantastic, even though I'm a CAR T proponent. And I think it comes back to our patients are going to need all of these things or a combination of these to really get to that cure. And until we get that cure, it is sequencing of them, and they've been phenomenal. So my patients who've had CAR T that now I need something else, we've had the bispecifics to go to, and they've done really well with it. So we have, so bispecifics technically are T cell engagers. So they're kind of like monoclonal proteins, but they have two, handcuffs, I like to call them. One is for the myeloma, either through BCMA, just like some of our CAR Ts, or with talquetamab, GPRC5D. So now we have another target. And there's actually a third one called FCRH5, cevostamab. So again, now we have 3 targets that we can actually go after in myeloma, which is pretty phenomenal. And then the other target is to CD3, which is getting the T cells that are already in your body. So we call them endogenous T cells. So as this protein's floating around, it's handcuffing all these things together, bringing them together. And then, as the T cells kill, they bring other white blood cells, et cetera, to help kill more of that myeloma. So the mechanism is a little bit similar to CAR T, but it's just instead of taking cells out and growing them and putting a receptor directly in there, you're kind of passively doing it in the blood. But again, it's the T cells are already in your body. So we think of CAR T having much higher response rates for the most part, but the bispecifics as single agents have been in the 60%. So they're still pretty high. If you think about, DARA 10 years ago, the response rate was 30%. So it's still, as a single agent, double that. But it probably, because we're not increasing the number of T cells and we're not directly going, it's still 60%. And I think, they're given by shots, most of these that are already elranatamab, teclistamab, and talquetamab. But you have to give them continuously because there's not as big of an expansion that you see. So as of right now, that's how they're given, either once a week or once every 2 weeks. We're hoping that in the future we can do fixed duration, which with cevostamab we have. You only give it for a year and then you kind of follow up, and hopefully we'll have data on that soon. But I think the toxicity is also a little bit lower when it comes to CRS and ICANS. We don't see as much, high grade CRS. Really, 70% of patients will get a fever, but we don't really see the oxygen dropping or anything like that. Same thing with ICANS. We might see the headaches, but not anything major. However, the big difference that we do see, because it's continuous dosing, is infections. So I always tell my patients, when you're on continuous bispecifics, when you're back to your homes, you're getting this with your local doctor, any fevers or any infection symptoms you have, really talk about it. Don't pretend it's a cold, because sometimes there are viruses like CMV or other viruses that we really need to help treat those pretty quickly. So I will say they've been phenomenal. It's off the shelf. I don't have to worry about a month or 2 months to make the cells, which for a lot of my patients, it's really needed. But again, there's a little bit of a difference in the infection rate, the high rate infections in the later part.
And then talquetamab, the GPRC5D, has a little bit of a different side effect profile as well. And certainly you've probably heard there's a lot of buzz around patients. So describing that, but then also some of the mitigation and management strategies as well.
Yeah. So I'm sorry. I was just talking about BCMA, and then GPRC5D, because it's also on skin cells and endothelial cells. So it can be on the tongue and nails. Basically, we actually see a little bit less infection with that, which is actually good. But we do see rash, for a lot of patients. We'll see really dry skin, like really bad eczema. People's nails can get pretty involved and actually even fall off if it gets really bad. So yes, we have these mitigation strategies. All my patients get bottles of CeraVe or some kind of non-fragrant, creams to use, and make sure they're lathering up and their skin staying hydrated, and also staying hydrated, drinking water. But then we use clear nail polish. We're using different kinds of zinc. So a lot of different centers are doing different things that hopefully we'll be able to prove really helps decrease this, and then, give these options to everybody. But I think the mitigation, strategies do work. So we are seeing more patients that have an improvement in those, and then maybe they get grade 1, 2, and then goes down. But there are some patients that can get severe taste changes and weight loss. So the weight loss is the one big problem. Everything else we can kind of figure out and, decrease the dose. You give it once a month instead. We do these things and it works. But when people get a lot of weight loss and they can't gain it back, then that's where we have to stop and make sure we help them gain their weight back before we continue it. And there's some studies going on that too, as to, what is that mechanism, why some people lose weight and others don't. But again, if you're losing weight, some of my patients here are like, I'm excited. It's almost like Ozempic. I'm losing weight and my myeloma is treated, which I get. But at the same time, at one level, we need to kind of reverse that. We can't have it keep going. And so it's something really important to talk to your doctor about.
So this was brought up a little bit earlier, extramedullary disease, and certainly that falls into the high risk category. Dr. Martin, how are, I guess, both from results from IMS, but in general, treating extramedullary disease, seeing some more, seeing it emerge too after CAR T, and how to manage those patients?
Yeah, that's still an unmet need for treatments in patients with relapsed refractory myeloma. At the IMS meeting, we saw an update from Yale Cohen based on combining the two drugs, combining teclistamab and talquetamab, the GPRC5D plus the BCMA. And actually those data represent the best data that we've had for treating EMD. We can't do that right now off the cuff because those two drugs are so expensive. We're not really allowed. The insurers won't pay for it. But in fact, the results are the best for that study. So they combined GPRC5D plus the BCMA bispecific. And in the patients that had EMD had a response rate that was over 60%. And at 12 months, still over 50% of the patients were still in remission. Those are the best data that we've seen for sure. Right now, if I'm treating somebody that has EMD, I would be okay with treating EMD based on a bispecific or a CAR T cell therapy. Maybe I would prefer a CAR T cell. It's an individual patient, decision. But that said, my usual course of action, if it's EMD that's approachable by radiation, I usually will give radiation concurrently with these therapies because I want to try as much as possible to sterilize that area. But there's also maybe some thoughts that if you radiate the area, it might open up some of the areas in that tumor ball that actually will allow the T cells and allow the therapies to better penetrate that area. The thought is that in EMD, there might be some wall that you can't get through with some of these therapies. And that's why that ends up where patients actually have residual disease after the therapy or relapse in that area. So it's still an unmet need, I'd say. There are also new therapeutics. So the next generation iberdomide is called mezigdomide. So Paul Richardson has always showed data at a lot of our meetings at mezigdomide as a therapeutic that can actually get into some areas of EMD. And I would love to have combinations of mezigdomide with bispecifics for these. So I think, we know it's an unmet need. We're going to get at, what's the best therapy, but we're still in the research phase of that.
So kind of wrapping up this area, we talked about treatments that are already commercially available, the CAR Ts, the bispecifics. Could each of you comment on some of what you found most exciting, kind of that next generation of immunotherapy coming down the pike?
Well, I'll go first and I'll leave Krina to wrap it up. So the one thing I want to mention is, because we haven't talked about it much, is the DREAM7 and the DREAM8 studies. And these studies were using belantamab, the BCMA targeted ADC, together with pomalidomide in dexamethasone, and I would say that's the DREAM7 study. In terms of the DREAM8 study, it's with pomalidomide in dexamethasone versus Velcade. So it's bortezomib versus belantamab or daratumumab versus belantamab with a doublet. And belantamab won out in both scenarios, doubled the PFS. With both of these triplets, the PFS is likely to be over 3 years. In these studies, the belantamab, just like Krina said earlier for the frontline study, it starts off every 4 weeks, but it's going to quickly go to every 8 weeks, and it might even go to every 12 weeks. And that's going to be the only injectable that you're going to need once every 3 months. That's not bad for a therapy where you have to come to the center for an injection every 3 months. That's a lot easier, in fact, than a CAR T for some people, or a bispecific. And like Krina said before, there might be less infectious risk with this. I think there's going to be a population that bispecifics are going to be best for, a population that CAR T cells are going to be best for, and a population that belantamab is likely going to be best for, where there's lower risk of infection and lower need to actually go to the treatment center to get that infusion. So I'm excited to see further studies with belantamab.
Yeah, and I'll just end with, I think the combinations we've alluded to, but we have trials that are ongoing. So some of these trials were presented in terms of these are future trial or trials that are happening. We don't have data for, but we have CAR Ts against GPRC5D now. So can we get that same result with a different target, but hopefully less side effects is sort of what we're looking for. Another one and done. There's combinations with CAR Ts and bispecifics, either in bridging, using a bispecific with a different target followed by CAR T, or as consolidation. And again, just making sure we can do this safety-wise, but also can we now really get everybody into an MRD undetectable state where they can do really well. And then the cell mods with CAR Ts and bispecifics as well. We're really excited to see some of those results. And again, a lot of these studies are going in earlier line, and then, CAR T versus transplant. So cilta-cel versus auto, or in first line. I think we're really excited to see the data come out from that.
And so one last question that came in, which makes a lot of sense. First, we talked a lot about moving some of these therapies up to first line, but we didn't really talk too much about, or really at all, about smoldering and the idea of moving a bispecific or CAR T up to smoldering myeloma.
Yeah, that might take an hour. No, I think it's, I think the high risk smoldering patients who truly have, are likely going to have myeloma in the next 2 years. Yes, we don't want to miss where they end up with a fracture or end up with renal failure or something like that. Those are patients we really want to do something about. I think we need data to make sure that the toxicity isn't higher, because their immune system usually is still a lot better intact compared to patients who are really relapsed refractory. But yeah, if you can do a one and done and they don't need treatment for 10 years, I mean, that would be fantastic. We don't have that data. So I think the idea is great. I think it's making sure you're picking the right patients that really need that treatment. And it wasn't that they're going to do well anyway without treatment, that they're going to have smoldering for a long time. And so again, I think we just need to make sure it's the right patient population.
Any comments, Dr. Martin, before we wrap up on that one?
Yeah, smoldering is a tough nut to crack, especially in the last 2 seconds. And I would say I agree completely. I'm not a person to start people with smoldering myeloma, right off on therapy. I don't take that lightly at all. I think until it's proven that early therapy is better, and also what the best early therapy is. I think with bispecifics, you might be able to get away with 3 to 6 months of therapy and then be off. CAR T, I'm not ready to jump into the CAR T just yet with smoldering myeloma, because they got a long lifespan ahead of them in general. And to risk any toxicity in that light is tough.
Well, on behalf of the MMRF, thank you both so much for joining us today, and everyone else who participated. I also do want to thank AbbVie, Adaptive, Baxter, BMS, Cure Magazine, Genentech, Janssen, Kite, Arcellx, Pfizer, Regeneron, and Sanofi. All of those companies, their funding, their support of our educational programming made this session possible. And for those listening who still may have questions, please feel free to reach out to our Patient Navigation Center. They're a team of former oncology nurses who will take your questions via phone, email, web form, however you want to be in touch. So with that, thank you again, Dr. Martin and Patel. Have a great day.
Thank you. Cheers.